Welcome to the Deep Dive, where we take those really dense research papers, the ones you need to know but just don't have the time to wade through, and we hand you the ultimate intellectual shortcut.
And today, we are jumping into something truly fundamental.
Absolutely. It's about life's most critical decision-making process, cell fate. We're looking at that foundational dance between control and, randomness.
Right. The one that dictates if a stem cell becomes a heart cell, a neuron, or if that process goes wrong and leads to diseases like cancer.
And our guide for this comes from a really cutting-edge paper in Nature Machine Intelligence.
That's right. It introduces a computational framework called scDiffEq. And at its core, it's using a powerful deep learning approach, something called neural stochastic differential equations, or neural SDEs.
Okay, so that's our focus for this deep dive.
Yeah.
How is computational biology getting us past just taking snapshots of cells to actually predicting their future?
And the key to that prediction, the real secret sauce, is accurately separating out the two forces at play. You have the drift, that's the smooth deterministic part.
The predictable path.
Exactly. And then you have the diffusion. That's the essential and often noisy stochastic element, the randomness.
It's a huge challenge because, biology is all about motion, but our best tools, like single-cell RNA sequencing, they just give us these static pictures.
It's like trying to figure out the plot of a movie by looking at 1,000 random photos from it. You can guess, but you don't know the forces that move the actors from one scene to the next.
So to make this relatable, we have to start with that classic image.
Yeah.
The Waddington epigenetic landscape. You, the listener, really need to visualize this. It's like a marble rolling down a big contoured landscape with all these valleys.
Mm-hmm. That's the perfect analogy. And those slopes and valleys, that whole topography, that represents the cell's internal gene networks and all the external signals it's getting.
So that landscape dictates the main path, the drift.
Precisely. It's the predictable push. It says, okay, you started up here, you're most likely going to end up down in that specific valley. That's the marble just following the path of least resistance.
The planned itinerary.
But here's a reality that this new model, scDiffEq, finally addresses. That marble isn't on a perfectly smooth track. The whole landscape is sort of vibrating, it's jittery.
So there's noise.
There's noise. That random vibration, that ground roughness, that's the stochastic noise, or what we call diffusion. And it's the force that can sometimes knock that marble over a little ridge and into a totally different valley, a different feat.
Okay, and this is where the big innovation comes in,
Yeah.
right? Because previous models, they understood the slopes, the drift, pretty well.
They did.
But they treated that vibration, the diffusion, as if it was a constant hum across the entire landscape.
Yes, exactly. A huge assumption. They basically said the wobble is the same whether the cell is, a stable, committed heart cell or a really flexible, undecided progenitor cell.
Which just doesn't feel right biologically.
It's not. And that assumption was a major roadblock. Biology tells us that noise has to vary. A cell that's plastic, that has tons of options, it should have more internal noise, right? More diffusion, especially when it's sitting at a decision point.
So scDiffEq is the first model built to capture this dynamic, this state-dependent balance between the deterministic and the stochastic.
It's the breakthrough.
All right, let's get into the computational side of this. We know cRNA-seq destroys the cell, so all we have is a single point in time. A position on a landscape, we need some kind of inference to figure out the rest.
And if you look at the older tools, you can see their limits. Trajectory inference is kind of just, it's guesswork. RNA velocity makes a lot of assumptions about how fast RNA is made and degraded.
Even the really sophisticated models like Prescient.
Even Prescient, it's a great model, but it was fundamentally limited because it treated that diffusion term as homogeneous and isotropic.
Even uniform.
Uniform in all directions and constant across all cell states. It was just a single number you had to tune for the whole data set.
And scDiffEq shatters that by using these neural SDEs. So can we walk through how it works without getting totally lost in the math? What are the key pieces?
Okay, so the model is trying to learn the forces acting on a cell at any given moment. And here's the genius of it. It uses two separate deep neural networks to learn each of the two forces.
Two networks for two forces. Okay, let's start with the predictable one.
That's the drift term, which is written as 5ZZ. This is learned by the drift network. Its entire job is to look at the cell's current state, its gene expression, and calculate the slope of the Waddington landscape right at that point.
So it tells the cell, okay, based on where you are, you're being smoothly pushed in this direction.
Exactly. The drift network learns the highways, but now for the revolutionary part, the noise.
The diffusion network.
Right, the diffusion term, or raw slade. This is learned by its own dedicated diffusion network. And because this network also takes the cell's current state, C size, as an input, the noise it outputs, its magnitude, becomes explicitly state-dependent.
So the model actually learns to turn up the wobble when a cell is at a crossroads.
And turn it down when the cell is stable and committed. For the first time, it's not just passive noise. The model learns that the cell is actively modulating its own internal instability.
That feels like a huge conceptual shift.
It is. The final equation just combines that predictable push, 5 at $, with that state-dependent wobble dimber, which then gets multiplied by some pure randomness.
So if the model is simulating all these cell journeys, how do you train it? How do you make sure its simulations are biologically, real?
They use a special kind of loss function designed to compare distributions. It's called the Sinkhorn divergence.
Which is based on something called the Wasserstein distance. I've heard it called the Earth mover's distance.
That's the best way to think about it. It's a great mental hook.
Okay, so help us visualize that.
Imagine your model simulates a bunch of cells and predicts where they'll all be on day 4. That's your pile of dirt.
My predicted pile of dirt.
Right. And then you have the actual experimentally observed cells at day 4. That's where the dirt should be. The Wasserstein distance is just the minimum amount of work, the least shoveling cost, needed to move your predicted pile of dirt so that it perfectly matches the real pile.
So by minimizing that shoveling cost, the model learns the right forces, the drift and diffusion, that actually move cells from the start point to the end point correctly.
That's it. It's not just guessing correlations. It's reconstructing the forces that drive the whole process.
Let's do a quick recap here.
Sure. So recap 1, mechanism and innovation.
First, the big idea. scDiffEq realizes that biological noise, or diffusion, isn't just constant static. It's actively modulated by the cell's state.
Second, the solution. It uses 2 separate neural networks, one for the drift, which is the predictable movement, and one for the diffusion, the state-dependent noise.
And finally, the training method. It minimizes that shoveling cost, the Sinkhorn divergence, to make sure the predicted cell populations match the real ones over time.
Which brings us to the big question, does it actually work better?
Right, how do you even test this? The ground truth problems is still there.
This is where the researchers got access to an incredible data set. It's called Larry Lineage and RNA Recovery. It tracks mouse blood cell development hematopoiesis at 3 different times. Day 2, day 4, and day 6.
And what makes it so special?
Heritable barcodes. Every cell was labeled with a unique barcode that got passed down to all its descendants. So for the first time, they knew the actual experimentally verified ground truth of which cell at day 2 became which cell type at day 6.
That's the gold standard. Okay, so task 1. Cell fate prediction. Could it predict where a progenitor cell would end up better than the previous state-of-the-art?
Unequivocally, yes. The biggest version of the model, scDiffEq, hit a fate prediction accuracy of 58.5%.
Now, 58.5% might not sound amazing in, say, computer vision.
But for predicting something this complex, it's a massive leap forward. It's a 7.8% improvement over the previous best method, prescient. That's huge for this field.
An almost 8% jump in predictive power on something as chaotic as blood development.
Yeah.
That is enormous. And did they also find a kind of sweet spot, an optimal tuning for the system?
They did. They tested the relationship between the 2 forces and found that performance was best at a specific drift-to-diffusion ratio, or $2 of $2.05.
So the deterministic push needs to be about 2.5 times stronger than the stochastic wobble
to get the most realistic and stable, yet flexible development. It's amazing that you can boil down such a complex biological process to a number like 2.5. It suggests development operates under some pretty tight rules.
But the real proof, the smoking gun that this new approach was the key, came from the ablation study.
Yes. This is the crucial experiment for everyone to grasp. They compared the full scDiffEq model to a version where they literally just removed the diffusion network. They forced it back to that old assumption of constant uniform noise.
So what happened? What was the cost of turning off that cell-specific noise?
The model it fell apart. Removing that cell-specific diffusion caused an 11.1% drop in prediction accuracy, a massive hit.
Wow.
And qualitatively, the broken model could only produce these oversimplified, strictly monofate trajectories. It just couldn't capture the multipotent nature of the cells. It forced them down one simple path.
So if you ignore a cell's ability to tune its own chaos, you completely lose the ability to model biological choice. That's definitive.
It is. And it also nailed task 2, which was interpolation. They trained it on day 2 and day 6 data, hid the day 4 data, and asked the model to predict that missing middle. It reconstructed that unseen intermediate population significantly better than its competitors, which means it's not just guessing the start and end points, it's accurately modeling the entire process of getting there.
Okay, let's do another recap.
Recap 2, performance validation.
First, the prediction leap. scDiffEq hit 58.5% accuracy in blood cell fate prediction, which is a 7.8% jump over the state-of-the-art.
Second, optimal dynamics. The best performance happened at a drift-to-diffusion ratio of 2.5.
And the big proof point. Removing that state-dependent diffusion network caused an 11.1% drop in accuracy, proving that self-specific noise is essential for modeling complex development.
So now we get to the really powerful part, the generative modeling. Because scDiffEq learns the actual governing forces, you can start running what-if scenarios.
You can do experiments in silico on the computer.
Exactly. They ran a virtual gene editing screen. They took a group of key transcription factors, genes known to be involved in making granulocytes, and they artificially cranked up their expression in the day 2 progenitor cells. Then they just hit play and simulated for 4 days.
What did the simulation predict?
It predicted a clear dose-dependent shift in the cell populations. The neutrophil fraction, a type of white blood cell, jumped from about 30% to almost 48%. At the same time, the monocyte fraction dropped from 28% to 14%.
That's incredible. It's the difference between saying gene X is correlated with this outcome and saying if I boost gene X by this much, I can engineer a specific quantitative increase in this outcome.
And it captures the nonlinear effects that simple models totally miss. It also validated known, complex biological relationships.
Like what?
Well, they looked at the famous rivalry between 2 master regulators in blood development, SPI1 and GFI1. The model correctly showed that SPI1 was broadly expressed, as it should be, across both paths. But GFI1, which is a repressor, showed sustained, highly specific expression only in the cells fated to become neutrophils.
So it's recreating known biology at a mechanistic level. It's modeling the fine-tuned control switch.
Precisely. But for me, the deepest biological finding comes when they linked all this back to cell plasticity. They grouped the progenitor cells by how many different fates they could produce. Some were monofated, some bifated, and some were multifated, producing 3 or more cell types.
Okay, so what did they find about the forces controlling those really flexible, multifated cells?
They found that the maximum strength of both the deterministic push, the drift, and the stochastic chaos, the diffusion, was highest in the multifated progenitors.
Wait, that's a little counterintuitive. I'd expect high diffusion, sure. More wobble means more chances to fall into different valleys. But why the highest drift? The highest deterministic push?
That is the critical insight. It tells us that when a cell is at a complex crossroads, it doesn't just sit there passively wobbling around. It actively cranks up its internal machinery to create high momentum, high drift, and high sensitivity to noise, high diffusion.
So that's maximizing its ability to quickly commit to any of the available paths.
Exactly. It's a strategy to maximize flexibility and minimize the time it spends in a dangerous, unstable state. Plasticity requires both controlled, high-powered movement and high chaos.
That really does redefine biological noise.
And finally, the tool proved it wasn't just a one-trick pony, it generalized. It worked on complex human hematopoiesis data, and crucially, it also worked on the kind of data most labs have, a single snapshot in time.
How can it model dynamics from just one snapshot?
They used another method, cytotrace, to infer a developmental timeline, a pseudo-time, from that single snapshot. And once they had that time axis, scDiffEq could successfully map out the dynamics. It showed it's not limited to these super expensive, hard-to-get lineage-traced data sets.
So it can work with most of the data that's already out there.
It can, which is a huge deal.
Let's do our final recap on this section.
Sounds good. Recap 3, applications and insights.
First, generative power. It successfully predicted these non-linear dose-dependent cell fate shifts from in silico gene experiments, and it validated known regulatory switches.
Second, the deep biological insight. Plasticity is an active state. The most plastic, multifated cells have the highest levels of both drift and diffusion.
And finally, broad applicability. The whole framework generalizes across species and, importantly, to single-snapshot datasets, making it incredibly useful for the wider research community.
So when you pull it all together, scDiffEq is really this new platform for probing cell fate decisions. It finally lets us disentangle the deterministic from the stochastic.
And the main takeaway, the thing to really remember, is that modeling that state-dependent diffusion isn't just a nice-to-have feature. It's essential.
Absolutely. The old constant noise models were giving us an oversimplified, almost cartoonish view of development.
But the paper does point out some limitations, right?
Yeah.
It's not a perfect system yet.
No, and they're very upfront about that. The main bottleneck right now is that it relies on a pre-computed PCA latent space.
A standard way to reduce the dimensions of the data.
Right, but when you compress tens of thousands of genes down to just 50 dimensions, you might be losing some of the fine-grained mechanistic details. The model might get fooled by correlations in that compressed space that aren't actually causal.
So future work needs to bring in more types of data.
Exactly. Things like protein levels, chromatin accessibility, maybe even move toward different algorithms. But the foundation they've built here is incredibly solid.
This research really does reframe how we should think about all those little fluctuations inside a cell. It's not just error or wobble, it's an actively regulated dial that's necessary for flexibility.
The cell is choosing its own level of chaos.
So that brings us to the final provocative thought for you to consider. We've just established that cells actively control their internal noise, their diffusion, as an essential strategy for generating diversity and flexibility.
An active, necessary component.
So if this randomness isn't a passive error, but a vital tool for creating healthy diversity, how might that change our approach to diseases like cancer, where cell fate control is just disastrously lost? Is cancer's uncontrolled nature due not just to a broken deterministic drift, but also to a completely dysregulated stochastic diffusion? Maybe the key to restoring order isn't just to stop the car, but to fix the suspension, to control the noise.
A profound shift in perspective, moving from just trying to suppress the noise to maybe selectively regulating it.
Something to think about until our next deep dive. Thanks for joining us.